Hello everyone, this is going to be for Biochem 2. This is going to be that pyruvate transition into acetyl CoA. This is going to be just the lecture notes, and the other video will be just steps of this pyruvate conversion to acetyl CoA. So let's get to the lectures that the professor gave us. Okay, so to get to acetyl CoA, in order to bring that to fruition, acetyl CoA it's produced in the mitochondria. So how do we get the pyruvate to the mitochondria? So we would have to transfer the cytosol pyruvate via the translocase. Alright, so then we could also get to acetyl CoA, also known as acetate. It's a 2-carbon molecule. We could also use fatty acids and amino acids as well. And then, again, you're going to then make the acetyl CoA, which we're going to go over the steps of. And then cellular respiration is the process by which pyruvate from glycolysis is oxidized into 6CO2 and H2O. When you have this full oxidation, you're going to have this larger increase in released energy in ATP. This is a very gigantic negative delta G when you're doing the full oxidation. There are 3 stages, and the last stage would be the electric transport chain, or oxidative phosphorylation. Alright, let's talk about acetyl CoA production. Again, you're going to be making ATP, NADH, and FADH2. Carbs will release 1/3 of the total potential CO2 during stage 1. Alright, let's talk about the acetyl CoA oxidation in the citric acid cycle. This is going to produce more NADH, FADH, and 1 GTP. GTP is basically ATP because you could convert the G to the A, so you can make ATP in the citric acid cycle. However, it's only 1 per turn. However, again, taking into account there are 2 glucose molecules. Oh wait, no, sorry. There's 2 of these acetyl CoAs. It's going to technically be 2, but per acetyl CoA in the citric acid cycle, you're going to make more NADH, FADH2, and 1 GTP, basically ATP, 1 ATP. Alright, oxidative phosphorylation, we'll be using those electron carriers for more ATP. Alright, so now, let's talk about the locations. In eukaryotes, stages 2 and 3 are in the mitochondria, so that means the citric acid cycle and the oxyphosphorylation, which is step 3, also known as the electron transport chain, are going to be in the mitochondria. The citric acid cycle will be in the matrix. However, the electronic transport chain, that will be in the inner mitochondrial membrane. Actually, succinate dehydrogenase is an enzyme that's in the citric acid cycle. However, it's also complex number 2 in the inner membrane of the mitochondria, so it's kind of the only one that isn't in the matrix. Okay, so pyruvate to acetyl CoA, the net reaction will be the oxidative decarboxylation of pyruvate, and they're going to be the first carbons of glucose to be fully oxidized when we make this CO2 in the process. This is going to be catalyzed by the pyruvate dehydrogenase complex, and you're going to have 5 coenzymes. There's going to be bound prosthetic groups such as TPP, lipoic acid, and FAD, and you're also going to have co-substrates of CoAsh and NAD. The first step will be pyruvate with this prosthetic TPP on enzyme 1, and this process will make HETPP, hydroxyethyl TPP, and CO2. And the enzyme 1 of the complex is a dehydrogenase. The second step, we're playing around with the enzyme 1, the dehydrogenase, as well as the transacetylase. And in this step, you're going to have the oxidation of HETPP. You're going to also have the acetylation of the lipoic acid. Lipoic acid will usually be in the oxidized state, and then it becomes acetylated, and then it gets reduced. We're at the stage where we're going from the oxidized lipoic acid to the acetylated form. And then so after that, in the next step that we're going to go over, you're going to see that lipoic acid become reduced, but then we have to restore it. So again, step 2, oxidation of HETPP and the acetylation of the lipoic acid on enzyme 2. So this kind of has enzyme 1 and enzyme 2. Enzyme 1, again, is the dehydrogenase. Enzyme 2 is going to be the acetyltransferase. Sorry about that, I kind of messed up the name there. I called it something else. It's actually enzyme 2 is the acetyltransferase. Alright, step 3, you're going to be making acetyl CoA. You need CoAsh here. This is the step where you need the you need the co-substrate CoAsh in step 3, where you're going to be making acetyl CoA. And again, enzyme 2 that you'll be using here is acetyltransferase. Number 4, remember you're talking about we need to regenerate the lipoic acid on enzyme 2 into its oxidized form. This is where we're going to do that. We're going to be using FAD in number 4. And we're going to make FADH and the oxidized lipoic. This is going to be restoring that lipoic acid on enzyme 2. We're going to be using a second dehydrogenase for enzyme 3. Alright, so now we need to restore our FAD, our oxidized FAD. Right now, after step 4, it's in its reduced form of FADH2. We need to get back to FAD. So, therefore, using that same dihydrolipomide dehydrogenase, we're going to be taking NAD, and then we're going to be reducing it into NADH. However, since it's a dehydrogenase, that's enzyme 3, remember, since it's a dehydrogenase, we're actually going to just strip the hydrogen off of the FADH, and we're going to just give it to the NADH. So now we have restored our FAD in its oxidized form, and then after step 5, we've also had the oxidized lipoic acid restored on enzyme 2. Okay, so CoAsh, let's talk about some of these different substrates and prosthetic groups. So CoAsh, again, it's this co-substrate. It's not a permanent part of the structure. They fulfill a function and then they dissociate. This one carries the small carbons. This carries acetyl groups. CoAsh carries acetyl groups. You have this very important thiol group in the molecule. That is HS. That's the reactive area. And again, it dissociates off of. It's a co-substrate, CoAsh. All right, let's talk about lipoic acid. It's also known as lipolysine or lipoamide. And this is a prosthetic group again. Prosthetic groups are tightly bound. Our other prosthetic groups are TPP and FAD. There's going to be a covalent link with the lipoic acid to the enzyme via lysine. This lysine has the amino group. Yeah, let me see if I wrote that correct. Yep. Yeah, lysine has this amino group on it. So you need a lysine there for this covalent linkage. Okay, so you're going to also have the oxidized form. Again, we were talking about the different states. This is the state where it starts often. Lipoic acid will be oxidized when you have the S bond to the S on its molecule. And then you become an acetylated form. Remember how we were talking about step 2, where we're kind of having enzyme 1 and enzyme 2 working together, and we have this acetylation of lipoic acid? Well, that's where we're going to get the acetylated form of lipoic acid. We have an SH and the acetyl group, instead of the oxidized where it's just S bond to S. And then after step 3, unfortunately, we're going to have our reduced lipoic acid, which is going to be SH and SH. Again, we need to restore this reduced form back to the oxidized form where it's just S and S. And this is done via step 4, where we're taking FAD and a dehydrogenase, and that FAD becomes FADH2. And then that will restore or oxidize lipoic acid to the S and the S. All right, let's talk about the PDC structure. So the structure of the complex itself, it's 10 megadaltons. It's very large. It's a multi-enzyme complex. There are 3 enzymes in it that are together. You have the pyruvate dehydrogenase as enzyme 1, and then you have your dihydrolipoyl transacetylase. It's a transacetylase. And then you have enzyme 3, which is another dehydrogenase. The different coenzymes and cofactors that are needed, again, pyruvate dehydrogenase needs the phosphatic group TPP. Dihydrolipoyl transacetylase needs the lipoic acid phosphatic group, as well as the CoA co-substrate. Dehydrogenase, the third enzyme, will need our FAD, which is again a prosthetic group, and then our NAD, which is a co-substrate. You have this short distance in a multi-enzyme complex. So this short distance is very important. There's a short distance between the active sites, and this will allow for channeling of substrates from one site to another. There won't be many side reactions due to how short the molecule has to transfer over to the other catalytic site. The regulation of activity of one of the subunits will regulate all. So we have a very important concept of regulation. You regulate one of these very short distance catalytic sites, it regulates the whole enzyme complex. Okay, now it's just the reaction again. This is an overall reaction review of it. So step 1, again, decarboxylation of pyruvate to the aldehyde. You're going to need TPP as well as the dehydrogenase enzyme 1, and you're going to make CO2 in step 1 of this process, as well as HETPP, hydroxyl ethyl TPP. All right, step 2, we have the acetylation of the lipoic acid to form a carboxylic in the lipoic acid. So basically, remember our lipoic acid on enzyme 2, it was the oxidized state, the S and S. Now it's going to be SH and then S and then this acetyl group. So we're going to have the acetylation of it. Enzyme 2, remember, that's our transacetylase. So the transacetylase will be playing a role in step 2 and step 3, more prominently in step 3, where we have CoA, our co-substrate, will come in with the enzyme 2, and we're going to make our acetyl-CoA. However, we're going to unfortunately reduce our lipoic acid in the process, and again, we need to restore that reduced lipoic acid that has SHSH to just SS. How we're going to do that is, again, step 4, we're going to take the reduced lipoic acid and FAD. FAD, again, was one of the prosthetic groups. When we have our reduced lipoic acid and the FAD, and we use the enzyme 3, the dehydrogenase, this is going to oxidize the lipoic acid. The hydrogens are going to go on to the FAD to make the reduced FADH2. So now we've restored our lipoic acid by step 4. Step 5, we're going to now restore or regenerate the oxidized form of FADH2, which is FAD. So we're going to regenerate FAD via the reaction NAD to NADH plus FAD, and that's going to, again, include the third enzyme, the dehydrogenase. All right, that's going to be all of the kind of lecture notes for the pyruvate complex, the PDH complex, and the steps and all that. However, we do have regulation. So regulation, it's going to just be the common sense stuff. Low ATP, high NADH, and high AMP. That's going to just drive the PDH complex forward. The PDH complex also has this thing where if it's phosphorylated by a kinase, this means it's inactive. There's a PDH kinase that will phosphorylate the complex, and that'll cause it to be inactive. However, if it's not phosphorylated, because a phosphatase came in and stripped away the phosphate groups, the PDH phosphatase, when it doesn't have the phosphates on the PDH complex, this means that the whole complex itself is active and it's going to, again, be pushed forward. All right, so that was a little bit of regulation on that, and we also covered all the steps briefly and the other extra lecture notes on it. All right, thanks so much. Please do something nice for someone, and best of luck.